Now we are at the right time. And it is my pleasure to introduce Resia Pretorius from the Stellenbosch University that will talk to us about the impact of microcirculation and microclots in ME/CFS. Thank you.
Good morning, everyone. Thank you very much for this opportunity to speak to this wonderful group of individuals, researchers and patients, and also to the online participants. Thank you very much for this opportunity to speak to you. I will be taking you through, in this presentation, through a whole process. But first, I want to thank many of my collaborators, most listed here, some of them not listed here, a wonderful team of students that's always been in my lab, collaborators from all over the world, and some of them really working very hard currently also to figure out long COVID specifically. And then I would like to thank the wonderful funders. I cannot do this work without Polybar Research Foundation, Bolvi Research Foundation, and many more. Really a very warm thank you from all of us. So I'd like to take you on a journey which started probably around 2007. And during that time, I was working on coagulation, and we were studying clotting pathologies, platelet hyperactivation, red blood cell structure, and it progressed where we worked on various diseases, various methodologies used. Particularly, we focused on flow cytometry, proteomics, scanning electron microscopy, and all sorts of markers to study coagulation. So coming to 2020, we were well prepared for this new pandemic that hit the world. So our interests are to look at how platelets interact with each other. They are potent signaling entities, how they interact with each other when they become hyperactivated, during inflammation and circulating inflammatory molecules that cause them to hyperactivate, and how they also interact with other types of cells of the immune system, red blood cells, and also the endothelial layers, where they might form complexes when they are activated in the presence of inflammatory circulating molecules. Then we were also interested in how the molecule, the clotting molecule, fibrinogen, interacts when they are exposed to any circulating inflammatory molecule. In the case of platelets, the interactions are through various receptors, where the receptor is activated by the inflammatory molecule. In the case of proteins, there's a direct protein-protein interaction, and this protein-protein interaction might cause the actual protein biochemistry of the molecule to change. And this change causes the protein then to form plates instead of single soluble molecules and to turn insoluble. And specifically fibrinogen is very capable of interacting with spike protein, LPS, and other molecules. The protein-protein interactions bind to the structure and the whole protein unfolds. And we discovered probably around about 2014, 2016, in that period, that when this interaction happens, the fibrinogen molecule turns into an amyloidogenic type of molecule. So the structure becomes plate-like, and instead of becoming or being a soluble molecule, the molecule turns out now to be insoluble little structures in circulation. Now, our early work in 2016, we first published how this the molecule LPS from bacteria can interact with the fibrinogen, and we used the marker, the green marker that you can see on the screen, called theoflavin T. Now, theoflavin T is a molecule marker, quite an old marker, that was first used to look at the amyloid nature of plaques in brains of Alzheimer's patients. So this well-known old marker together with Congo red. So we decided to look at it for the first time when we exposed fibrinogen, and we used both from plasma in circulation or purified fibrinogen. We exposed that to LPS and then exposed this mixture to our marker, theoflavin T, and it lit up like Christmas trees. So what happens is when the LPS, or the inflammatory molecule, binds to the fibrinogen molecule unfolds, and it opens damaged hydrophobic areas on the molecule. And that allows the theoflavin T marker to bind to it. So this process is then called an amyloid binding process. And we then discovered that fibrinogen can become amyloid. It's not the same thing as amyloid beta in the brains or alpha-synuclein. It's not the same thing, but if you do have a brain slice of someone with Alzheimer's with amyloid beta, syn-alpha-synuclein in Parkinson's, you can use theoflavin T, and it will bind into the areas that has amyloid formation. So we also looked at stroke blood way back then, and here you can see the typical little spiky little fibers in blood, which is the amyloid nature, and in A is of a healthy individual without any of these amyloid present. Now, way back then, we were also looking at thromboembolic stroke, and we were specifically interested in red blood cells. We also looked at what happens to hemochromatosis, but we were mostly interested in the red blood cells and the platelets. And the clotting pathology we knew, but we didn't deep dive too much into that at that point in time. And I'll fast forward and I'll show you in a few slides what happened when we went back to our old micrographs and looked at it again in 2024, 2025. So fast forward to the pandemic, we were well prepared. We knew about amyloid. We knew about platelet hyperactivation. We knew about red blood cell damage during infection with LPS, circulating inflammatory molecules. What we weren't prepared was to see the absolute havoc that was happening in the blood of patients in the ICU. We found platelets significantly hyperactivated. Look at the yellow arrows on the slides. It looks as if the platelets just exploded. Red blood cells were interacting with platelets, but we also found that there were huge amounts of structures, of deposits, of aggregates, of plasma protein that even glue together red blood cells. And you can see them in the blue, with the blue arrows. So quite early on in the pandemic, we also tried to see what happens when we added spike protein. There is part of the spike protein. We looked at microfluidics, and we could indeed, in a healthy individual, induce these platelet hyperactivation as well as microclot. We didn't call it microclots formation then, but aggregates of certain of the plasma proteins. We could induce it simply by adding spike protein to whole blood and to plasma from a healthy individual. We also looked at the microfluidic system, compared the controls to the controls with spike protein, and with COVID-19, we saw huge activation of the structures, these aggregates, just due to the spike protein. So that gave us the idea why in the whole blood of the ICU patients, we found significant deposits on our red blood cells, platelets being hyperactivated. And we had a basic idea that in that stage, it was mostly due to the spike. So fast forward to long COVID, our first cases in South Africa was probably around about the end of 2020, after speaking to Amy Prohl from Polybio Research Foundation. Myself and Doug Kell, my long-standing collaborator, I had a discussion with her. She said we must look for samples in South Africa. We went out and we did find them. And lo and behold, the white blood cells, red blood cells, platelets significantly damaged. And we also found these deposits, aggregates in the plasma, stained with theoflavin T. Here's just an example of a very ill patient that we saw when we traveled to Germany, to a clinic in Germany that treated patients with long COVID with HELLP apheresis. So fluorescent microscopy of long COVID showed exactly the same patterns. We found these deposits, the green deposits once again. We have marked the red blood cells here with red blood cell marker for the membrane. And you can also see the platelets hyperactivated in pieces of broken platelets everywhere. And then scanning electron microscopy, close-up of red blood cells of long COVID patients we saw these deposits on and around the red blood cells. Now you can imagine if someone's blood looks like that, the red blood cells will not be able to carry oxygen properly. It will not be able to get the oxygen into the cells where it's needed, and you will get little areas of the endothelial layers that might die, especially in the capillary beds, because they don't receive enough oxygen. Now, in 2021, we quickly did a proteomics analysis after we started having our first long COVID samples. To our surprise, we could not break down the plasma of the long COVID and acute COVID samples, but we could break it down with trypsin from controls, plasma from controls, and diabetes that we included in the study. So that gave us a thought that perhaps these deposits, these plasma deposits, the clotting deposits, do not break down as it should. And when we did develop a protocol to be able to break it down, we found various molecules entrapped inside these structures. Now, interestingly, again, Amy Prohl from Polybio, we had a discussion with her, and she said we should not call these deposits what we call them. We called them heterogeneous, dense matted amyloid deposits. So she said just find some different, easy flowing term, and we came up with the term microclots. And from then on, these deposits were now known as microclots. And entrapped inside these microclots after digestion, and we've done proteomics, we found one molecule of interest, alpha-2 antiplasmin. And that is a molecule that blocks the breakdown of pathologies, clots, microclots or clots, inside the blood. So it stays there for a little bit longer, and then it can cause havoc that we have been seeing specifically in long COVID. We quickly realized that we cannot only use a macroscopy method for a quick analysis, and also we can't use proteomics as a regular point of care type of test, just too expensive and too time consuming. So together with various of our collaborators, patients, clinicians, parents of patients, family members, we crowdfunded, but with, again, Polybio Research Foundation and Kernels. And we could have our imaging flow cytometer in our lab within 8 months after starting the process. And we developed a method and published on it how to find microclots in platelet-poor plasma using an imaging flow cytometer. What that means is typically the flow cytometers are used for cells. It's what they've been developed for. However, we could actually develop a method to track microclots inside the plasma, which is not necessarily cellular of nature, but these aggregates of plasma proteins that is insoluble. So let's look a little bit more deeply into what it means for the patient. We believe that one of the things that can happen if these deposits stay in circulation for quite a time is that ischemia reperfusion injury can happen. So in general terms, what is this ischemia reperfusion injury? It is initiated via bursts of reactive oxygen species produced during the reoxygenation phase following hypoxia. So this is one of the many of the complaints that patients have, that they have this ischemia reperfusion injury. And we believe that central to this is the presence of these microclots in circulation that do not break down as they should, and then actually block the tiny capillaries. Recently, we've also been working with a team from INSERM and Montpellier under the lead of Alain Theory. He's a world expert in NETs, but in cancer. And he contacted us a few years ago, and we just had a paper where we showed that these microclots are actually entrapping nets from neutrophils as well. So it's a little immune complex in circulation. And there's an example of one of the micrographs that we could machine learn also from our micrographs that we took under a fluorescent microscope, where we could see that there are DNA interactions with this, the microclots stained with THT. He also did ELISAs elastase assays and myeloperoxidase ELISA assays. So it's a whole lot of markers pointing to immune net activation and that they are jet-trapped and structurally associated with our microclots. Now, is a microclot, or should we actually be focusing on their content, their activity, and their biochemical characteristics? So over the past year or so, our team has been specifically focusing on that. We, with proteomics, found numerous inflammatory molecules, including fibrinogen, all the fibrinogen different chains in circulation, but we were now also the past months deep diving in what we can see about the inflammatory molecules that are entrapped in the clots. And we used a program called ameliogram because we are interested in amyloid protein. So ameliogram is a molecule that you can put a sequence of your protein into this program, and it can determine what the amyloidogenicity of your molecule is. So how sticky it is, how damaging it is when it's in circulation and when it interacts with fibrinogen. Here's just one example, and I turn again back to alpha-2 antiplasmin. This is our molecule that prevents clot breakdown. If you look at the amyloidogenicity, and this is the gram, the ameliograph that is produced, you can see that the dotted line there, all of those sequences above that shows high amyloidogenicity. So we looked through all of our molecules that we and others have found in proteomics, and we have determined that most of the molecules, the inflammatory molecules, spike protein is another hugely amyloidogenic protein. Sophie Nystrom and Per Hammerstedt discovered that in Sweden a few years ago. All of them have got a huge potential to turn molecules that they interact with, but they themselves are amyloidogenic. So if they are amyloidogenic, it means they are sticky. It means they will be in circulation. It means it will cause hyperperfusion injury and damage to the vasculature and drive inflammatory processes. So the next step that we are interested in, that we have been doing, and we've just submitted a preprint on this, is what are the different types of microclot phenotypes? We know they are heterogeneous, and we know they are complex. So we think they are seeding onto debris from damaged blood cells, vessels, cell membranes, immune complexes. We know that they are associated with nets. They can be fibrinogen alone, just interacting and folding on itself because of binding with spike protein. And they can also seed onto blood cells that are still intact, so functioning, but not working as they should. So what do we think what's happening? We think that what is happening is when you have damage is by circulating inflammatory molecules, the membranes of white blood cells, red blood cells, endothelial cells become prothrombotic, and they can become debris. So this debris can be in circulation. It happens in healthy individuals as well. However, in a healthy individual, your immune system works well, and your immune cells will get rid of them. In patients with ME/CFS and long COVID, we know that the immune system is muted. So our immune system doesn't get rid of the debris as it should. And these debris parts of breaking down cells just stay in circulation longer than they should, with a muted immune system, a prothrombotic nature, the PS flip or phosphatidylserines flip happening on the membranes, causing them to just become huge little traps for pro-inflammatory molecules. And then the procoagulant and amyloidogenic nature of these molecules cause them to form the seeding areas of these of fibrinogen, as it's prothrombotic, serum amyloid A, which is another hugely amyloidogenic molecule in circulation. And these little areas are then one of the types of phenotypes in circulation that we believe are a huge problem, because the debris of cells breaking down are not removed from our circulation. But then the microclot complexes, as I alluded to in the beginning, can also just seed onto intact cells, although these cells may not be functioning as they should. And here we have seen that they can attach to, this is micrographs on confocal microscopy as well as fluorescent microscopy. They can just attach to all sorts of cells, binding them together, blocking with a blocking capability where the blood doesn't flow as it should, and can actually cause problems for oxygen transport and just functioning and blood flow in general. So, as I mentioned, one of the complexes is just where the fibrinogen molecule interacts with a inflammatory molecule like spike protein. So you have the conventional clotting cascade, but when these inflammatory molecules interact with these soluble fibrinogen molecules, you have a process where the fibrinogen molecule itself becomes insoluble, and it is a little prothrombotic little area without the canonical process of clotting. And obviously they can also be involved with thrombin, but then they form these dense matter deposits that can also be a clot that stays a little bit longer in circulation. So circling back to our work on stroke. So when we're looking back at our red blood cell work on stroke, we also we again saw these dense matter deposits. And in a recent publication with a whole team from University of Liverpool, University of Washington, we looked at actual stroke clots that was taken out or by just getting the little clot out of the brains of the stroke patient. We found that these stroke clots actually do also stain with theoflavin T, showing that a clot, an actual true clot in the stroke clot, a thrombotic clot, also has got a severe amyloid signal. Our latest preprint was at looking at post-viral disease, but also looking at vaccine injury. We did the proteomics, and it was interesting that if we look at fold changes, that the people with post-viral and vaccine injury split out well from those only with long COVID. So it was quite interesting for us to find that there are specific signatures, molecular protein signatures that differ between this group. Then we also looked at exercise-induced changes of microclotting together with cytokine levels with our collaborator Mark Foy from the University of Derby. So they did 2 CPET interventions, and we looked at the microclot profiles. And what's interesting for us was that it seems after CPET intervention of long COVID patients, we found that it seems as if the bigger microclots break down into smaller microclots afterwards, but they reform again. So we think that microclots are definitely on the pathway of disease. So our paper therefore challenges the rationale of graded exercise therapy, and it underscores that obviously we must be very careful in patients with ME/CFS and long COVID to just force them to do exercise randomly. We are also looking currently at a nanotweezer experiment, and this is a microclot manipulated by a nanotweezer, and we are trying to look how we can manipulate these microclots and bring them together and draw them out and break them up with a nanotweezer. So this is quite interesting work that we will be doing with our physics department. And then our latest findings, and I'm not going to share too much because the paper is coming out preprint soon. We have deep dived into the actual what's happening proteomic level, but at sequence level. And the thing that I can say is, together with my team at Stellenbosch and samples from Canada, from POTS and long COVID, we found that we've got post-translational changes on our fibrinogen and other molecules that relates to glycation and glycation end products on certain sequences. And that's a huge thing for us, because we think now we might really start to understand the amyloidogenic nature of the molecules. We have also published a paper where we tried to give some sort of methods for clinicians to use, and we discussed how they can search for endothelial damage in this paper. Then, very interesting that we think as a simple test that a clinician can use is nail fold videocapillaroscopy. You look at the nail fold and you can see the rarefication of the vasculature on the nail fold. We think it's a simple way. It's not the be all and end all, but at least you can look if there's some endothelial damage and thrombotic endotheliitis in your patient population. Another very easy method to be used by a clinician in the practice is infrared imaging. So one can see that infrared imaging reveals the effect of the impaired vasculature. It's a quite old technique. It's used quite frequently in diabetes, where you can actually see the hot and the cold with infrared areas in the body, suggesting that there are damage and that the blood is not flowing as it should. And then the last thing that I want to do and talk about is the development of a point-of-care microclot detection and characterization device. We call it the MDCD. So this device was built by one of my MEC engineering students, and we know that clinicians definitely not, and many of the researchers do not have access to a microscope. And a Zeiss microscope, the one that we have, costs a lot of money. We built this with a fraction, 1% of the cost. And you can see it now is still very much a prototype. But if you look here, this is a microclot, and my student Luanais Palmer spent quite a time on this. She validated with theoflavin T and exposed to microclots or to plasma. She detected the same, she could detect the same microclot on the Zeiss and her MDCD. So her MDCD instrument uses also the various fluorescent type of processes to detect that the Zeiss does. The Zeiss obviously have lots of channels. With our MDCD at this stage, it's just the green channel because we are interested in the theoflavin T. But then I also asked her recently to just look if we could find PAC1 FITC, which is also the green, staining using our MDC, and she did. And look at that. I think the MDCD is actually picking up better than the Zeiss. So what is PAC1 FITC? PAC1 FITC binds, it's the marker, the fluorescent marker that binds to the activated receptor that binds fibrinogen. So it means that if this is, for example, in your circulation, and this is a part of a piece of debris, this part of microclot forms then part of the cellular debris component or complex, and that there is, for sure, FITC, PAC1 receptors for fibrinogen. But what's so interesting as well, as this specific receptor do not only bind fibrinogen, it also binds the hugely amyloidogenic serum amyloid A, the exact same receptor, binds that molecule too. We and others have found that serum amyloid A is significantly increased in patients with long COVID, and also I think in ME/CFS. So it means that the prothrombotic nature is not only driven by damaged fibrinogen and amyloidogenic fibrinogen, it is also most definitely driven by circulating inflammatory molecules like serum amyloid A, which we know is the mother of all amyloid proteins. So we hope with this work we will be able to take this work further. My student is now completing a master's degree. We are going to go with this into a PhD if she gets funding for it. Tomorrow evening at the Polybio Research Conference, if you would want to pop in at Amy Prohl's conference, Luana is going to go into depth about her MDCD and to explain exactly how she does it and what she did to make this instrument work. So we hope in future we will be able to have a very, very low cost, real proper fluorescent microscope at a fraction of the time that people can use. What is needed in long-COVID and post-COVID research? As we all know, we need to stratify phenotypes. We think if we can use fibrinogen, if we can use amyloidogenic nature of microclots, in somewhere in the process, we can at least stratify the patients and group those together that in fact do have a prothrombotic nature and is more prone to clotting abnormalities. We're very excited that we are working with wonderful teams all over the world that is trying out various methodologies to look for microclots. Jeroen Den Dunnen's group is doing the same work as Mike van Elzak has worked with PET scans, also where they look for fibrinogen in the body and onto the vasculature. We hope that those research efforts will really tell us. Sorry, can you hear me? I think I'm off. Can you hear me now? So that those type of research will really show us what is happening to the vasculature and how clotting pathologies are impacting patients with suffering from long COVID. We have patented our method early in 2021. So the microclot testing method is actually available now in some places overseas where we actually test patient samples at selected pathology labs and we use imaging flow cytometry. And this is some of the places, and we have a new collaborator in the UK, PeploBio, that will also be very soon start using the test in the UK as well. The problem with that obviously is still a imaging flow cytometer method. However, Biocode Technologies, our company, is working very hard to try to optimize the method for a normal flow cytometer. And I must report that we have done it successfully so far. We are co-staining with various markers. So we're not only using theoflavin T anymore, and hopefully those type of methods will be available very soon for patients all over the world. This is my wonderful team. I cannot do my work without them. Some of them sitting there at the back. It's really a team of passion. We like to work very hard and see where our research take us. And we hope that sometime in the future, these type of tests that we can produce in our lab will be available for every single patient, not only for selected few. Thank you very much.